Welcome to the class. Topic for today is micropropagation. Micropropagation is defined as the clonal propagation of vegetatively propagated crops. It is the process of multiplying plants by stock plant material. By this procedure, a large number of progeny plants can be multiplied and then they can be planted in the field condition, and all these plants which have been multiplied by micropropagation would be similar to the mother plant. Micropropagation has been divided in different stages. So number one stage is initiation stage, also known as establishment stage. In this stage, first of all, a piece of plant tissue, which is also known as explant, it is cut from the plant firstly, then it is disinfested. Disinfection is done to remove the surface contaminants which might be present on the surface of the explant. For this explant is washed with a mild detergent like 2 in 20. Then it can be treated with an antibiotic or a fungicide depending upon the chances of infection which may infect this particular plant. If it is infected by a bacterial species, then it can be treated with an antibiotic. If it has more chances of infection for a fungal species, then it can be treated with a fungicide. And then it is surface sterilized with a sterilant. The sterilant may be any of your choice. It may be hydrogen peroxide, ethanol, mercury chloride, sodium hypochlorite or calcium hypochlorite in varying concentration. After the disinfection, the explant is inoculated onto the medium. Inoculation is a process when the explant after surface sterilization is placed onto the nutrition medium. Nutrition medium plays a very critical role in the process of micropropagation. This medium provides all the nutrients which are required for the plant cell for its growth and development. For this, the medium contains mineral salts which are required for growth, a carbohydrate source, mostly a sucrose, and a solidifying agent such as agar are present in this medium. The objective of the stage, the initiation stage, is to achieve an aseptic culture. Aseptic culture is one without contaminating bacteria or fungi. So, establishment of a culture that is free from any type of contamination and is in growing stage is known as the achievement of initiation stage. That is the first stage of micropropagation process. Moving to the next stage, stage number 2, that is multiplication stage or sometimes known as proliferation stage. So a growing explant can be induced to produce vegetative shoots by including a cytokinin in the medium. The medium which is used here in the multiplication stage is also known as shoot induction medium. Shoot induction medium mostly having richer concentration of cytokinin. Cytokinin is a plant growth regulator which promotes shoot formation from growing plant cell. For shoot formation, the amount of cytokinin should be always higher if we are using it in combination of auxin. Most commonly used cytokinins in plant tissue culture medium includes BAP, that is benzyl aminopurine, kinetin, zeatin, etc. And these are adenine derivatives. After the multiplication of shoots, these well-developed shoots are transferred onto a fresh medium that is known as root induction medium, and this stage is known as rooting stage. So third stage of micropropagation is rooting or pre-plant stage. These growing shoots can be induced to produce adventitious roots, and for this these shoots are required to transfer onto a root induction medium which is having higher concentration of auxin. Auxins are plant growth regulators which promotes root formation. Auxins are indole derivatives and the most commonly used auxins are indole acetic acid, naphthalene acetic acid, indole butyric acid, etc. For easily rooted plants, an auxin is usually not necessary, and many commercial labs will skip this step. They achieve rooting in the ex vitro condition, that means in greenhouse condition, by potting the shoots. Stage number 4 or final stage of micropropagation is acclimatization, where a growing rooted shoot can be removed from the tissue culture vessel. Its roots are washed properly under running tap water to remove all the agar traces from the roots, and then it is placed in soil. The soil which we are using here for the potting of this tissue culture raised plantlet should be sterilized by autoclaving. When this is done, the humidity must be gradually reduced over time because these tissue culture raised plantlets are very susceptible to wilting. So if we summarize the whole stages of micropropagation, first thing that is very important is the explant source, or we know them as plant material or mother plant material. So it may be either these axillary shoots or it may be the apical shoots or shoot buds. So any of the explant can be taken which is having higher meristematic activity. And for the stage 1, that is establishment of explant, for this stage, this explant is transferred onto the medium where shoots are established in culture around 2 to 3 months. Then these shoots are transferred to the second stage, that is shoot multiplication stage, where the medium uses shoot induction medium, and there will be small clumps of shoots which increase in size and number of leaves are there, and it will take around 2 months. Then comes the third stage, that is the rooting stage. Rooting can be done either in vitro or ex vitro. After this, when plants are established, they are transferred into individual pots for 3 to 4 weeks under mist. After proper development of a plant, they are transferred to into individual pot and then they can be sold in the market or can be transferred into the field condition. So these are all the required stages of the micropropagation: establishment stage 1, multiplication stage 2, rooting stage 3, and last acclimatization stage 4. Now coming to the approaches to micropropagation, how micropropagation can be achieved in lab condition. It can be done either by axillary shoot or apical bud multiplication, or organogenesis, or by somatic embryogenesis. Axillary shoots and apical buds, they may be quiescent or actively dividing meristem. The axillary shoots are present in the axil of the leaf and apical buds are present on the apex of the branch. The axillary buds, which are located in the leaf axils, they are capable of developing into shoots. But in natural conditions, only a limited number of axillary meristem can form shoots. The most practiced method of axillary shoot and apical shoot proliferation is meristem culture. In meristem culture, apical meristem is taken as an explant and it is a dome of tissues which are located at the extreme tip of a shoot. The apical meristem along with the young leaf primordia constitute the shoot apex, and it is used for the development of disease-free plant, that is the most prime concern in case of micropropagation. Meristem culture is always used to raise disease-free or virus-free plant. For this, meristem or shoot tip is isolated from the stem by a V-shaped cut. The size of the explant is mostly 0.2 to 0.5 mm of the tip, and it is critical for culture. In general, the larger the explant, the better are the chances for culture survival. The explant is very small in size and it is very tough to survive in the tissue culture. This is the structure of apical meristem. The apical meristem has been divided into 4 zones. This is permanent tissue zone, which is having vascular bundle and pith, and cortex is also here. This is zone of differentiation, zone of elongation, and on the tip is the zone of cell division where maximum cell division is happening, having the highest meristematic activity. It is harboring the meristem tissues and leaf primordia is here. It is not having any connection with the vascular tissue. So if any pathogen is present here in the vascular bundle, since there is no development of vascular tissues, so this pathogen will not travel to the meristematic region. Hence, this explant will be free from any pathogenic infection and it is used to raise disease-free and virus-free plants. Organogenesis is the production of organs either directly from an explant or from a callus culture. Organogenesis depends on adventitious organs which are arising either from a callus culture or directly from an explant or on the formation of axillary bud to regenerate whole plants for some time of tissue culture. There are two types of organogenesis. First is direct organogenesis. In direct organogenesis, direct regeneration of the explant into plantlets can be tried. This has been achieved many plant species by altering the hormonal combination of the culture media. For this, the explant taken can be either axillary buds or shoot tip. From these explants, shoot induction is done, which is followed by root induction and finally acclimatization. In case of indirect organogenesis, there is one more phase that is added here, and that is the callus phase. So callus is first produced from the explant. Organs can then be produced from this callus tissue or from the cell suspension which is produced from this callus. So if we want to go for the cell suspension culture, that can be done from this callus, or adventitious shoots and then roots can be produced via the callus phase, and this procedure is known as indirect organogenesis. Next type of micropropagation approach is known as somatic embryogenesis. That is formation of embryo-like structure from the somatic cell. And these somatic embryos are similar to the zygotic embryos. That means they have induction of both. At the same time, root and shoot can be induced from these somatic embryos as happens in the case of zygotic embryos. First of all, somatic embryogenesis was practiced by Raynaud in the year 1958-59 in Docus carota, that is carrot plant. These are the different developmental stages of these somatic embryos. They may be termed as globular shape, torpedo shape, heart shape, and finally the cotyledonary shape, as like happens in case of zygotic embryos. In some cultures, somatic embryogenesis occurs directly in absence of any callus production from pro-embryogenic determined cells. Pro-embryogenic determined cells, PEDC, which are already programmed for embryo differentiation. So this is known as direct somatic embryogenesis. The second type of somatic embryo development needs some prior callus formation and embryos which originate from induced embryogenic cells, IEDC, within the callus tissue. So this is known as indirect somatic embryogenesis, where somatic cells are developed into the embryo-like structure, and this somatic embryogenesis is induced under in vitro conditions. There, two distinctly different type of media may be required. One medium for the induction of callus, and then the second media which is used for the subsequent development of these cells into the embryo-like structure, which are known as embryos. Advantages of micropropagation. Micropropagation is a high volume, large scale propagation method. By this, genetic uniformity of plants can be maintained and direct delivery of propagules can be done to the field. If we are making many thousands plants from a single explant, then we can lower the cost per plantlet. Rapid multiplication of plants can be done if once your protocol is optimized. The synthetic seed technology offers tremendous potential in micropropagation and also in case of germ plant conservation. However, future research is needed to perfect this technology so that it can be used on a commercial scale. What are the synthetic seeds? The structure which you can see here, these are the synthetic seeds. Synthetic seeds are also known as artificial seeds. These are encapsulated somatic embryos. As embryo is encapsulated, it is having a covering of endosperm and finally the seed coat for its protection. Likewise, these somatic embryos are encapsulated and they can be used as seeds and have the ability to convert into plant under in vitro and ex vitro conditions. These somatic embryos are bipolar structures with both apical and basal meristematic region, which are capable of forming shoot and root respectively. Based on the techniques, there are two types of artificials which are produced. One are desiccated synthetic seeds, which are produced naked, or polyoxyethylene glycol encapsulation is done. And second are hydrated synthetic seeds. Hydrated synthetic seeds are produced by encapsulating the somatic embryos in hydrogels like sodium alginate, potassium alginate, carrageenan, sodium pactate, or sodium alginate with gelatin. They are made by dropping method. Somatic embryos are dipped in hydrogel for encapsulation of somatic embryos. Hydrogels used may be any of the following. It may be alginate like sodium alginate, agar from seaweed, seed gums like guar gum, locust bean gum. Sodium alginate solution is prepared in MS basal medium. Then somatic embryos are dipped in this solution and these coated beads are added one by one into a complexation solution flask capped on magnetic stirrer and then they are left as such for around 20 to 30 minutes for the encapsulation. With these advantages, micropropagation has some limitations also. First, this particular procedure is equipment or facility intensive operation. For this to carry out, you have to establish a lab facility. You have to procure certain equipments to carry out plant tissue culture or micropropagation in lab. So it will cause some cost to you. Then you will need a technically expertise or skilled person to operate plant tissue culture or micropropagation. Then third condition comes, protocols are not optimized for all species. For some species where protocols are optimized, it will not take much time to perform micropropagation. But in case of the species where protocols have not been optimized, it will take some months to optimize the protocol. Then liners may not fit industry standard. For this, you have to go for certification. Whatever plants you have been producing, for that you have to get a certification from the government agencies. And if your protocol takes a lengthy time to be optimized and you cannot produce much plants from a single explant, then the cost per explant will be high and your plantlets will become expensive. One more thing that is a limitation to micropropagation is contamination. So when you are doing micropropagation, you have to maintain very strict aseptic conditions so that there is no chances of infection in your culture. Otherwise, many of the cultures can be destroyed by contamination issues. These are the books which you can refer for going through micropropagation. First is Introduction to Plant Biotechnology by Dr. H.S. Chawla and second is Introduction to Plant Tissue Culture by M.K. Razdan. And for detailed study, you can refer to this website, that is www.biologydiscussion.com. Thank you so much for listening this carefully. Thank you. Thanks a lot.